Exciting sunrise session. We've been doing this for a number of years, and they've been very successful. They're growing in popularity, as is evidenced by this morning's attendance. And I want to extend a very heartfelt thanks to our sponsor, Regents Blue Cross Blue Shield. And we have here with us today Kevin Bischoff, the Vice President for Business Development and Community Relations. Kevin, thank you so much.
Annette, thank you very much. I just wanted to say on behalf of Regents Blue Cross Blue Shield, thank you so much for coming. This is 7th or 8th year that we've been doing this now, and every single one of them have been incredible, and I'm sure that today's is going to be exactly the same. Utah State University is a very, very important institution to us at Regents. They're a very important client, a very important partner of ours, and a great university. And of course, a great university has great leadership, as epitomized by President Stan Ulbricht. Stan serves on our board of directors, and we're thrilled to learn from his expertise throughout the year. So please enjoy your breakfast and enjoy the presentation. Thank you for coming.
Thank you, Kevin. For those of you that don't know our president, this is Stan Ulbricht here, sitting beside Kevin. He's had a busy year. He's had a number of busy years at the helm of Utah State University, and the research you're about to hear today is indicative of some of the things that have been happening under his purview. During the past year, we, under Stan's guidance, joined together two important offices at Utah State, the Office of Graduate Studies and the Office of Research. As you can well imagine, the researchers are critical to a research institution such as ours. We're very proud of all the work they do, and we have very successful research going on at USU. This year we have had 12 Career National Science Foundation Young Professor Awards, which is phenomenal for an institution, and it's great. The bringing together of those two offices, graduate studies and research, required a person to be at the helm. And I'm very pleased to announce our new Vice President for Research and Graduate Studies, Mark McClellan. Mark, come on up.
Well, thank you all. Very happy to have you here. My wife Julie is here. We just moved here in July. Actually, Julie just arrived a few months ago. Selling a house in Florida is like selling snow to an Eskimo these days. And speaking of which, I hope you appreciate we brought some mild weather with us, although I know those of you who ski could not have that same opinion. We're looking forward to the white stuff coming in. In fact, we had snowshoes under the Christmas tree this year. So coming to USU was really exciting. It was an opportunity to step into a university that had a youthful vigor, an energy, and a sense of future. I've been at many universities. I worked at Cornell, Texas A&M, University of Florida, large, energetic, powerful institutions. But no place have I felt the sense of opportunity and the sense of possibility as we do here. And that speaks well of this institution, and certainly a very exciting part of my transition here. One of the things that was very attractive was the commitment of the state of Utah through the USTAR system to support leading edge research that would lead on to a partnership with the private sector in commercialization. It's just a huge investment, and one that our universities in this state can be very proud of and hold very dear, and really appreciate that investment. At USU, we're very proud to have that program under the leadership of Vice President Rob Buchanan. And Rob, would you stand up and take an appreciation for that, because it has been great. As Vice President of Research, is probably one of the most wonderful jobs in the university. It is also one of the most humbling jobs, because you get to be with and visit with some phenomenal, talented individuals that push the envelope and take you to a next level of thinking. In my history, I had the great honor of being on the faculty with Carl Sagan at Cornell, and you knew that individuals have the opportunity to change the world around you. And as I'm here to introduce this speaker tonight, Randy Lewis, you know that this is an individual that has the potential of changing the world around us. He's a great biologist. He comes to us with a lot of experience. He got his degrees at CalTech and then University of California at San Diego. He ended up working at some place called the University of Wyoming, and we were able to steal the heck out of him and convince him that the right place to be and the most exciting place to be will be right here in the state of Utah. I'm not going to steal his thunder, but suffice it to say, there's a reason that Slinkies are all over this room. Yes, that's what they are on your table. And so Randy's going to walk us in to the world of spiders, the world of silk, and the incredible biological potential of this material. Ladies and gentlemen, help me welcome Randy Lewis.
I tend to walk around, so I'm not going to be able to just sit in one spot here and do my thing. So let me get this one on. So is that loud enough you can hear me? Okay. All right. So I'll try to convince you that spider silk is the next super material, and in fact, it's a very ancient material. And it's been around for on the order of 350 million years. But I like to start out thanking all the people who actually do the work, because I'm long past being able to get in the lab, although I still am able to solve a few problems once in a while. And so this is the team we have now. Grab my pointer here. So I can, yep, there we go. All right, so I have three people who moved with me from Wyoming, Mike and Justin and Flo, our senior research scientists, and they were a big part of my team at Wyoming and are a huge part of the team at Utah State now. We have four undergraduates, Brianne, Andrea, Candace, and Howard are shown here. We have three more that were not here when we took this picture outside our brand new building at the USTAR Bioinnovation Center. And then we have two graduate students that have joined us here, Michaela and Cameron. So this is really the team that we have put together here. Then we have collaborators that we've got from other parts of the country. I'll talk about their work. Jeff and Greg are at ASU, and they've helped us with the physical analysis of the spider silk. Uris is a collaborator in Nebraska, and we'll talk about some of the work that he's done on how to spin fibers. Mac and Dawn helped with the spider silk conversion or transgenic silkworms, and then Holly and Heather and David were big parts of what we did at the University of Wyoming. Funding from USTAR, the Air Force, DOE, NIH, and NSF. I also have to thank Anna McIntyre because, as you'll see, the slides are much more artistic than I do. I passed art class when I was in high school only because I completed every assignment, not because they were good. So Anna took what I started with and generated something that looks much nicer. And finally, I do have to thank my wife because she left all of her friends in Laramie, moved to Logan where she knew nobody. She left a house that she designed, did all the decorating, all the landscaping, to move to a house that's sole redeeming feature was that it was along the Blacksmith Fork River, so that when I need to decompress, I can go out fly fishing. And if it were not for her buying into this vision of what we have for spider silk, I certainly would not be standing here today. So I definitely need to thank her. All right, so as was mentioned, there are really sort of two aspects to spider silk. And one is the material itself. The other is that people can understand it. People have walked into a spider web in the house and gotten it all over them. They're aware of spider silk. So I'm going to start with a little clip.
These? Yep, goats. These are our goats. So they're just regular
This
goats.
was,
They're
from the
absolutely regular
BBC,
goats.
and it
They're
came
not.
out
They're,
Wednesday night.
they're totally incredible goats. And so these kids have the genes for a spider in them.
Yes.
It's insane. And where does the spider silk actually come from? I mean, where do you get it?
Well, it was designed so it comes in the milk.
They look like such normal goats, but in fact they're totally unique and bizarre. I mean, this is bizarre.
I guess I would not say it's bizarre. I think that it's certainly different, but you know, they're absolutely normal. I don't think there's anything different about them. There you go.
So the pumps just go on like that.
That's all there is to it.
Oh, you can see it.
On there. Yep.
You can actually see it coming
Yep,
out.
you can see the milk coming out.
So this is exactly the same as any normal goat milking
Absolutely.
process.
Absolutely. Do it exactly the same. We can knock this open and you can take a look and see.
Well, just looks like normal milk.
Looks like absolutely normal milk. If you actually do an analysis of it and you look at all the components of the milk, the only thing you'll find is different is that there's one extra protein in there, and that's our spider silk protein. All the rest of it looks absolutely like normal goat's milk.
So that was a BBC. That was all shot here at the facilities at Utah State, and like I said, came out on Wednesday. This is a little clip from a NOVA program that came out last spring, spring of 2011. To just give you an
Spider
introduction
silk
to
has
the,
been shown to
the
have
silk
more
itself.
tensile strength than steel and Kevlar. It can stretch to 140% of its length without breaking and remains flexible even in extreme cold. It's also so lightweight that a mere pound of the stuff could form a single strand long enough to stretch around the equator. But could we ever harvest enough to put it to use?
All right, so to just sort of summarize, so spider silk is stronger than steel, stronger than Kevlar, more elastic than nylon, and on a weight basis is the strongest material that exists out there. And so if you compare it to the man-made fibers, the major difference is that you can combine elasticity and tensile strength. And that's something that none of the man-made fibers really fall into that category. So we'll continue on and try to explain where it is and why it's that way and how we do that. So why do we study spider silk? Well, I'm a basic scientist, and the idea is that we want to understand how do you make a protein. So spider silk is made up of proteins just like your hair, like your skin, and actually like most of your body. So how do you make a protein that's stronger than Kevlar and more elastic than nylon? Now, in order to convince somebody to give you money to do that, you have to say there's something coming out the other end for their investment. And our investment is that we're going to create new biomaterials. And in particular, we're looking at materials that can be used for artificial ligaments and artificial tendons, particularly to be able to bridge tears to allow for recovery. So what happens now is that in the recovery process, you usually have to immobilize them to allow them to heal. And when you do that, scar tissue forms. So what you need to do is you have to be able to move, to have movement for the ligaments and tendons for it to repair itself normally. We have materials, and I'll show you, we have materials right now that will allow for a bridge to be installed, that would allow the ligament or tendon to be used, at least in a limited way, to drive healing rather than scar formation. We have a lot of interest in terms of things like parachutes and parachute cords, with the idea that they're much more strong and they're flexible, so that when you dump a tank out of a C-130, about a third of them hit the ground at terminal velocity because the parachute collapses. We can prevent that from happening because the tank will basically cause extension of the parachute rather than collapse. We've actually been contacted by the people who are making the sails for the next America's Cup. Because what they'd like to do is every time you tack, of course, you have to move the sail. And when you do, you lose all of the energy that the wind put in your sail. But if that sail bows out when you turn, it's actually going to help make the turn because it's going to flex all the way through. Now, we obviously can't make enough, but I'm thinking they got enough money. We can probably talk them into paying a lot of money to help us make those. Airbags. You know, right now, the reason you don't put your kids or grandkids in the front seat is because an airbag is designed to blast you back in the seat. It's an explosion. Literally, it's an explosion. What manufacturers would like to do is do exactly what a web does. That is, keep something from going through by absorbing all of that energy rather than counteracting it with a blast force. All right? And finally, for sports apparel, as well as a number of different kinds of sports equipment like climbing ropes, we've got people who are going crazy thinking about climbing ropes, and you'll see when we talk about the details why it is that climbing ropes would be a big deal. So that's sort of an introduction into why we're studying spider silk. Now, the spiders that we study are an interesting group. They make the typical orb web. That is the round web that they make. It turns out that they've become evolutionary masters at materials. So they actually make 6 different kinds of silk in 6 different glands. All right? And if we look at those, we'll sort of quickly run through them. The first is called major ampulate silk, and it forms the framework of the web and the line that when you see a spider fall, that's what it catches itself with. Minor ampulate silk makes a little spiral in here, and they use that to know where to put all the rest of the components on the web. A cynniform is swathing prey, so they like to eat live prey. So what happens is that if they catch more flies or whatever it is in the night, they wrap them up to keep them from escaping, and then they come back for their midnight snack, and they know that they still have it alive. Okay? So instead of cold pizza, they have live flies when they get up for their midnight snack. They also use it to cover the eggs. It's called an egg sac on the inside. So it's a very soft place to put their eggs down. The next is piriform silk. They use it like, it's not really cement, it's more like lashing. And so for those of you who are Boy Scouts, and I honestly don't know whether Girl Scouts learn lashing, but I know Boy Scouts do. You know, the idea is that you can tie things down by just putting a lot of different kinds of a rope to tie things down. And that's really what they do. So when they're climbing along, if you watch, you'll see them, they stop, and then all of a sudden they do this little quick movement with their abdomen. What they're doing is putting this silk down just like rock climbers put pitons or chalks in, so that if they fall, they only fall to that point. Okay? So they just set them up, and so when they're crawling across the ceiling, they'll stop, they'll put this down so that when they fall, they'll only fall from where the last place they attach themselves. Flagelliform silk is actually the majority of the web. It's what they physically stop the prey from coming through. And then there is no sticky silk. They actually put a glue on. So the glue is called aggregate silk, and those are little droplets of glue on there. And the final one is really a tough silk that they put on the outside to protect their eggs. Now, we have cloned and sequenced the genes for every one of these silks and the glue. So we know what each of these silks look like. Now, there are 3 in particular that are of interest, and that's the dragline, the minor ampulate, and the flagelliform, because they have very, very different mechanical properties. And you can see that here. So I didn't put the numbers up there because the numbers won't mean a lot to except for a few of you. But dragline silk is very, very strong. It's basically the equivalent of Kevlar in terms of strength. It has moderate elasticity, and so it has a term that's called energy to break. Now, in the real world, energy to break is the most important thing. If you're a rock climber and you fall, when you hit the end of the rope, you are not weight, you are energy. So you don't care if the rope will hold 100 pounds of weight. What you care about is how many joules will it absorb before it breaks, because you're moving and you are energy. So in most applications, and it's the same for your ligaments, it's the same for a parachute cord, it's the same for a seat belt, for all of those things, it's energy to break that determines the usefulness of the material. And you can see that in energy to break, it's actually 3 times stronger than Kevlar is. Now, on the other hand, minor ampulate silk has lower strength than dragline silk, but very respectable in terms of strength, but no elasticity. What happens is when you stretch it, it deforms. It doesn't spring back again. It just stretches. So it ends up with a moderate energy to break. Flagelliform silk has about the same tensile strength, but a very large elasticity. It'll stretch almost as far as a rubber band. And of course, in the web, that's terrific, because here's the web, an insect hits, and the web just keeps giving and giving and giving until it's absorbed all the energy, and then the glue sticks it. So that's why very few things make it through a web, because the web just keeps giving and giving and giving until there's no more energy left. So it also has a very high energy to break. So just comparison to some natural materials here to show you that this really is a group of pretty astounding natural materials. Now, when we got done looking at all of the sequences of the proteins, we sort of started to see some ideas about, well, this part of the protein is only found in elastic silks, and this part of the protein is only found in very strong silks, but we didn't understand how that really worked. All right? So together with our structural people, what we did was analyze the structure. And so the first structure we found looks like this. And this is really, when it was turning, you can see it's 4 molecules, protein molecules, as they would be found in the fiber. And what you can see is they stick so closely together, there is no space in there. There is so little space that not even water can penetrate. All right? Now for the spider, that's great, because it means that when you set up a web, every time it rains or it dews, the web doesn't dissolve and go away. All right? So if you learn nothing else today, you can go home and tell your kids and grandkids that you know how spider silk works. Because this slide and the next two will explain everything you need to know. All right? And it's really easy. So what we have are Legos. Okay? That is, each of these molecules packed together with little pins and little holes, just exactly like Legos. Okay? And when you pull on the ends of Legos, they don't come apart. Now, if you pull sideways, and this is important when we come back here later, okay? If you pull sideways, they come apart. But if you pull them lengthwise, they stay together. All right? So that is where the strength comes from in spider silk. All right? So about 300 million years ago, spiders started playing with Legos. All right? Now, what's interesting is that they actually one-upped Lego, because they put pins and holes on both sides as opposed to pins and holes on opposite sides. And in addition, they put them in 3 dimensions. So it's as if now you start pins and holes on the side, so now you can stack not just in 2 dimensions, but in 3 dimensions. And that's what you see here, is a 3-dimensional Lego stack. All right? So the second thing we found was that, in fact, the elasticity comes from something that looks exactly like a Slinky. Okay? So when you stretch the silk, it stretches just exactly like a Slinky. And when you let go, it retracts. All right? So for 150 million years, spiders have been playing with Slinkies. Okay? So again, far before anybody else thought of a Slinky. Now the final component is a zipper. Now, we don't completely understand why they need a zipper, but we think what happens is that you can't put something very stiff and something very flexible together without some kind of a linker. And so what you get is a zipper, all right? And you can see here the teeth and how they fit together, all right? What's really amazing is that for 150 million years, the spider have never changed these teeth. So they have the choice of 20 different size teeth to put in their repertoire, and they use the same ones every time. For every species, for the entire length of every protein that uses, they use the same teeth for the zipper. Now, if you think about a zipper, that makes a lot of sense, right? I mean, those of you who do sewing know that zippers come in, what, at least 3 different sizes, and you can't mix them, right? You can't take the zipper that you have on your big heavy coat and put it together with a zipper for your pants, because the teeth don't fit. All right? So 150 million years ago, spiders learned that if you want a zipper, you better make sure that the teeth fit and are the right size. Okay? Now, one of the problems is that you'd like to just be able to farm spiders like you do silkworms. Okay? Silkworms are great. You take a couple thousand of them, you throw them in a box with a bunch of mulberry leaves, and you come back 14 days later, and you have a bunch of cocoons. You can't beat it. And that's why silk is cheap, at least in real terms. Spiders have three serious deficits when it comes to that. The first is they're territorial, okay? They don't like to hang around with each other. And secondly, they're cannibalistic, so they have a solution to that problem, and that is they eat each other until everybody has enough room. So it was tried actually in England and Britain in old farmhouses back in the 1860s where they would in fact put a bunch of spiders in there. What they found was they'd come back and there were many fewer spiders than they started with, but everybody had enough room to eat. Now I showed you that the web is actually made up of 4 different kinds of silk that have very different mechanical properties. So if all you did was collect the webs, what you end up with is, and apologies to the administrators, you end up with a committee designed by administrators to solve a problem. And that is, you have the whole diversity and you end up with nothing that works. Okay? And that's what you'd end up with if you combined all of the facets of these different materials. Because you can't have high strength with something that's very, very elastic because they don't mix correctly. All right? So how do you go about making large amounts of the material? So what we did was we devised a way to make the spider silk proteins and then spin the fibers. So I'll tell you a little bit about the different production methods that we have. So we're really working on 4. Bacteria are great. I can come up with some strange concoction that I want to make as a spider silk protein, and we've made 36 of them, by the way, most of which are not represented in nature. And in about 2 to 4 months, you can actually have bacteria with a new spider silk gene in it, making the protein that we spin into fibers. The problem is that scaling it up is enormously expensive. So if you took the biggest vat for production of bacteria right now, you could produce about 12 kilograms per run, and you could probably only do it a couple of times a week. And it would be an expensive proposition to be able to do it. It's just not a practical sort of way to do it. So about a decade ago, we combined with a company that's now gone defunct, and not because of the spider silk, because they had another project that the Department of Defense paid $500 million to get a hold of, so they didn't worry about the spider silk anymore. And we transferred, as the little clip I showed you, a spider silk gene into a goat, and that milk, the protein is produced in the milk. And you can produce about 18 kilos per goat per year, and it takes about 1 to 2 years to generate a whole new set of goats. But you can breed them up very, very rapidly to be able to expand a herd to whatever you want. Now, the nice part about goats is that if we want to use them for ligaments and tendons, we have a system that the FDA has already approved. That is, you can take proteins from milk, use them for drugs, and the FDA will approve it. So that's one of the major reasons why goats are an ideal system for at least that application. Alfalfa is another one. We saw that was mentioned in the little video. I was born and raised on a farm. Back in the days when you didn't have something that picked up the bales for you, you actually had to pick them up by yourself. But we have a system now for growing large amounts of alfalfa, for harvesting, transporting, using. And the nice part about alfalfa is that it makes lots of protein and it makes big proteins, which is what we want because the spider silk proteins are very, very large. Now, the other good thing about alfalfa is that when you're done taking our little bit of the protein out of there that's ours, we can turn around and use the rest of it in dairy to feed the cattle and pay for all of our expenses. Okay? So we can pay for processing by selling the waste product because all of the waste product is what you want to feed anyway, minus a little bit. And I'll talk to you about silkworms because honestly, once you have transgenic silkworms, you can basically scale up to whatever size you want. You can produce absolutely, if you wanted to, you could take over the world silk market if you could figure out a way to get people to buy it on that kind of a scale. So I'll run through each of these to give you a little bit of an idea about where we're at. So talking about...
I need you to take a look at what I pulled from the body bag.
This is from a CSI New York program. They came to us and asked us all how to do this.
Silk.
This is
On
not how we do
steroids.
it.
A spider that produced a strand this thick would have to weigh 150 pounds.
Exactly, so I ran into DNA for a species
There
determination.
is no DNA in the
A
silk,
spider
by the way.
has floppy ears, he bleats. Likes to eat garbage.
You lost me.
It's from a goat. Would you mind explaining to us
So the
what
goat
you're doing
standing
with this
on the
goat?
lab bench,
Transgenics.
not how
We
it's
spy
done.
spider DNA into the genome of a goat.
And you
Goat
can't
produces
just
silk
spin it
and
out of the
milk.
milk like
Simple.
this. You see them
One of
just
the
directly
strongest
spinning the
materials in the
fiber.
world, 5 times the strongest
If we could
steel.
do that, trust me,
Exactly.
we'd already be rich, okay? There's no question about it. It just doesn't work that way, all right? So the other thing about it is that the person who ends up being convicted of the murder is the lab director of this lab. So one of my best friends is at North Carolina State. For some reason, he's watching this and he's sending me, so they're at 2 hours ahead of us. So we know the program's coming out, so he's emailing me about the program. You know, and about halfway through, he goes, Randy, things aren't looking very good for you. And then there's a couple other things, and so the last email I get is you murdering SOB. And so, you know, I don't know what's going on in the show because I haven't seen it yet, but that's the end text, you know. So, well, this is really looking good for me, right? So, but this video clip, it was shot by the National Science Foundation.
This is a
And
story
this
about
one will
silk
give you how it
and
really is
milk.
done.
The silk is from golden orb weave spiders.
There you go, pumpkin.
They're incredibly inquisitive. They're a lot of fun.
The milk from specially bred goats. Good luck trying to connect those dots. So what's the thread?
There's a lot of interest in spider silk fibers because they're stronger than almost any other man-made fiber, and they also are elastic.
Because it's stronger ounce for ounce than other materials, there are many possible medical uses, from artificial ligaments to sutures for surgery.
So the question is, how do you produce large amounts of the material?
Spider farms just don't work. They tend to kill each other. So molecular biologist Randy Lewis figured out how to put the spider's silk-making genes into goats.
What we did was put that gene into some goats in a situation where they would only make the protein in their milk. And when the goats have kids, and then they start lactating, collect the milk, and we can purify that protein in much, much higher quantities.
With help from the National Science Foundation, Lewis studies spider silk at the University of Wyoming. And so far, Lewis says he's seen no differences in the health or appearance of the transgenic goats.
Good girl. Oh, come on. Come on. I know, you don't want to go down.
Feeding and milking goats and wrangling spiders are sometimes part of the job.
So we collect the milk out here, and then we take it back to the lab for processing.
The silk we're particularly curious about is the dragline. That's the outside of the web. It's the strongest part of the structure.
Chemical engineer Heather Rothfuss separates the silk protein from the milk. No arachnophobia for her. In fact, she's actually warmed up to working with spiders.
I'm on the roll now, so it's collecting okay.
Here's the silk
Just
racks
4
that's spinning
drops
ourselves.
of
You see it
protein
coming through
processed
here.
from the milk can be spun into 4 yards of silk.
So there'll be a lot of applications: eye surgery, plastic surgery, neurosurgery.
The lab is also introducing genes into alfalfa plants. So how do people react to this tangled web of a tail?
They understand that you can't just farm spiders, so you got to come up with another way to make the material.
No kidding. For Science Nation, I'm Miles O'Brien.
So no matter how we get from the protein from alfalfa, whether we get it from the milk, whether we get it from the bacteria, we have to purify the protein, then we dry it down, we redissolve it, and we force it through a very fine needle, and then you see the fibers that you form, okay? So that's the process that we use. So this is, just to give you an idea, so this is actually 100 strands of the fiber. So the finest fibers we make are about 5 microns in diameter. That's 100 fibers together. That would be plenty right there to use for an artificial ligament. That has plenty of strength right there to substitute for any ligament in your body. This is a thin film. So what we do for the thin films is instead of throwing it through a needle, we put it on some sort of a template, dried out. You can see it looks a lot like saran wrap. People are interested in it for coatings, particularly for things that are going into the body like stents, maybe also for various other sorts of medical devices. Because we know that this is, at least the natural material is very biocompatible. So it offers some real opportunities to be able to do that. Now, one of the problems that we encountered in terms of trying to spin the fibers was that we had very poor consistency. So here is force, so that's how much force it takes to stretch it. This is how far it stretches. And you can see that you have some that are really lousy, you have some that are not too bad, and you have some that are really, really good. All right? Now, if you're trying to make materials out of it, that's not a very good situation because you have, a twofold difference in how much strength it has. You can see we've got a 10, 15-fold difference in how far it stretches. So since we moved to Utah State in June, we've actually figured out how to solve that problem. And so here's an example of what happens now, and you can see that every one of the fibers, and there's only 5 of them here, the total study had 20, but you know what happens when you put all the colors together? I learned that in art class. What color do you get when you put them all together? Brown, right? So the only thing you'd seen was a brown line here, and you wouldn't have known anything, all right? So we only use 5 here so you can see that all 5 of them look exactly the same. If I put 20 there, you wouldn't have been able to tell anything in terms of that. So the problem is that when we get done with the spinning, they're still not very good. So what we learned was that if you stretch them in an alcohol water bath, they get much better. And if you then dip them in water and stretch them a little more, they get much, much better. So we looked at the structure of these, and we did it using both NMR, which is like MRI for small molecules, uses exactly the same principles, and X-ray diffraction, which is the same thing as taking X-rays, except again we're looking at small molecules, but the X-rays have much prettier pictures. The NMR doesn't have a very pretty picture. So this is what it looks like when we first spin it. So it's about 68 microns in diameter, tensile strength's not so good, elasticity really sucks, and overall toughness is not very good. What you see in the X-ray is this very pretty ring with absolutely nothing there, which means that you have no real structure. That is, this thing is amorphous, which means nothing there. All right? Now, if you put it in the alcohol water bath and stretch it, you see that the diameter shrinks, you get a very large increase in tensile strength, a pretty large increase in elasticity, and the toughness jumps up. And what you can see not very well is you start to see multiple rings, okay, which starts to indicate structure. So what we know is in examining those rings that what we did was we made the Legos, okay? So what happened here is there were no Legos. But once we put it in the alcohol water bath and stretched it, the Legos formed. Now probably because the individual proteins, when we first made it, couldn't figure out, didn't have the room or didn't have the flex to get everything together, okay? So now we have Legos. However, remember that the key to the Legos is you have to have them so that you pull on the ends, not pulling on the edges. So in order to do that, you have to make sure that these are parallel to the fiber axis. You want your Legos lined up parallel to the fiber axis, okay? And what happens is when you put it in the water, it reorganizes, and this very strong amount of X-ray diffraction right along the equator indicates that now not only have we made the Legos, but they're lined up parallel to the fiber axis, and now all of a sudden you see a much larger increase in tensile strength, a huge increase in elasticity. We're still not as good as the silkworms, but we're really in, or as the spider silk, but we're in very good shape in terms of looking at the structure. So if we look at these and make some comparisons, how good are we? Well, so this is some of our better materials, and what you can see is that compared to the ligaments and tendons, we have plenty of strength. In fact, we can make things 100 times stronger than a natural ligament. We can make things that are 10 times stronger than tendons are. So we have materials right now that we can use to make artificial ligaments and tendons or materials to help satisfy that problem. Now, if we compare them to some other man-made materials, what you see is that in terms of toughness, we already have materials that are better than Kevlar, carbon fiber, and high tensile steel in terms of toughness. What we don't have is we don't quite have the strength. So there are applications where we need more strength. But you can see that at least on the toughness end, we're ahead of the game. But unfortunately, we're still a long ways behind the natural materials. All right? So in toughness, we're getting close. Tensile strength, we're still more than a factor of 10 below, factor of 5 to a factor of 10 below. The reason that we think this is happening is bacteria only make short proteins, okay? That is, they choke when you make a protein that's very large. So we know that the proteins in the natural silks are very large. And we're making ones that are about 1/5 to 1/3 that size. We think that's the reason why we can't get the strength up. And then I'll show you some data to sort of confirm that in a second. Now, we tested with the collaborator at the University of Nebraska another way to spin fibers. It's called electrospinning. And it's really pretty simple. What happens is you take your solution, you put it sort of on one pole of a battery, and you put the other pole over here, and you just keep jacking up the voltage until finally, just exactly like you expect, a spark shoots across there, except instead of a spark, the silk protein shoots across there as a liquid. All right? Now what he learned is that typically what you see happen when you do that is this. Okay? You end up with a mat. All right? So what he learned how to do was to do the same thing, except that he's able to make something that is in parallel so that it looks more like this. So he ends up with something that is a parallel fiber. All right? So even though it's composed of lots of little teeny fibers, they're all parallel to make a yarn. Okay? And that's what you see some of here. So here's our 5-micron diameter right here. So that's the smallest we make. You can see that almost every fiber in there is way smaller than 5 microns. Okay? So we then tested them. What we find is that two sort of curious things happen. That is, as they get smaller, they get stronger. Now, one of the things you always do is you measure strength compared to the diameter of the fiber. Because we all know that if the same material was thicker, it's going to be stronger. So you always ratio it to the diameter of the fiber. So what happens is, in real terms, they get stronger as they get smaller. All right? Now, there's lots of materials that do this, okay? Not all of them, but lots of them. What's unique about spider silk is that as it gets thinner, it also gets more elastic. So there are no materials that our collaborator has tested that has both of those things happen with the same material. So some materials do this, but none do both. So what it means is that the material actually gets better the thinner we make it. Now there are a couple of other interesting things about spider silk. One of the things that's really interesting is that the faster you pull it, the stronger it gets. So if you pull it we normally pull it at a few centimeters a minute. If you pull it at 15,000 yards per minute, or per second, 15,000 yards per second, it's 3 times stronger than if you pull it at a normal speed. Which means that for very rapid applications, it's even better than it is under normal circumstances. Okay? Now, one of the other crazy things about spider silk is that it transmits heat as efficiently as copper. So those of you who are cooks know that one of the best things you can have is a copper bottom pan because it transfers the heat to whatever's inside of it very rapidly. Turns out that spider silk does that as well. And what's interesting is that it's even more efficient than copper when it stretches. Now, we've got no clue why that is, okay? But we're working on that. So there are a number of other things other than just the strength and the elasticity that imply some interesting applications for spider silk. Now, a couple of us were sitting around the lab and talking about 5 years ago, I guess, and said, you know, it's great to be able to purify the protein and spin the fibers, but what if we could generate an organism that would make our protein and spin it into fibers at the same time? And of course, we thought of silkworms. And so the paper just got published the beginning of January after an arduous journey through the peer review system, which took us almost 8 months to get it published. But what we demonstrated was that we were able to transfer a spider silk gene, in this case a very large spider silk gene, and a smaller version of it, into silkworms. They would make that protein in the silk gland only when they were getting ready to make their own cocoon, and they would spin a cocoon that contained our spider silk protein along with the proteins they normally use to make the cocoon. So I'll show you a couple pictures. So one of the things we did was that in order to make sure that we were getting it to work, we put a little tag on there that when you shine it with UV light, it made a bright green picture. So for those of you who are here in the flower child era, you remember the fluorescent posters, the black light posters? Well, this is our biological black light poster. So this is in fact looking at the silk glands without the UV light. You can see they actually show up almost green, but boy, when you hit it with the light, you see them flat. So what we knew then was that we were making the protein and we were making it in the right place. Okay? Then we asked the question, is it being made and put into the cocoon? and you don't even need the black light to see that. So here is the control cocoon. Here are the transgenic cocoons. You can see that they're all looking green-yellow. And again, if you hit them with UV light, they light up just exactly like the gland did. They become bright green. And you can see each of the little fibers in there and all of that sort of stuff. All right? So we had a system that made it. The problem is that we didn't have lots and lots of spider silk protein in there. We only had about 5%. But when we looked at the effect on the cocoon, it was actually pretty astounding. So on the top, this is the control silkworm. And these are lab-adapted silkworms. So these aren't the best silkworms in the world. They're just easy to work with in the lab. This is the smaller, this is 2 strains, the smaller version of the protein, and this is the bigger version of the protein. And this is real commercial silk. So this is actually taken from a Korean commercial company. So if we compare just in terms of strength, what you see is that compared to the control, our 2 smaller versions of protein are about 50% stronger, but our larger one is 80% stronger. And all of them, and this one is 50% stronger than the natural Korean. Now I'm not going to show the data, but it turns out that they're also about 50% more stretchable. So not only did we increase the tensile strength, but we increased the stretchability of the natural material. Okay? Now we're still a long ways from having spider silk, but we've got pretty impressive silkworm silk now at the very least. All right? So what do we conclude from all of this? Well, in order to make stronger and stretchier materials, we need longer proteins, as you saw in the silkworm, and we're sure that's going to be the same for the other ones. That you make smaller fibers, okay? So the smaller you make them, the better they're going to be. And finally, at least if we're going to use the artificial spinning system, you have to stretch them and do water treatment. And for every one of the 36 silks, well, actually there's only about 22 of them now that have been tested, but of all the silks that we've spun fibers from and tested, stretching makes them better, putting them in water makes them even that much better. So it's a very consistent sort of a thing. You need to be able to use the proper spinning conditions, and I showed you that we've developed those to be able to make very consistent materials. Then a little bit, about 5% of spider silk spiked into the silkworm, saw a very large improvement in the silkworm. And what we've run into that I never knew was the situation until we started getting serious about looking at commercial applications, and that is that 5-micron fibers are too small for anybody in the world to use for manufacturing purposes. So what we've designed is a multi-fiber spinning. So that is, we're going to spin 10 or 15 fibers together to make a yarn which is now big enough for U.S. and worldwide manufacturers to be able to use it. Because we went around and basically, you know, everybody sort of laughed when they said, yeah, we got these great 5-micron fibers. And it's like, who cares, because we can't use them. So we're going to put yarns together, and that will be the fiber that then gets made into textiles and used in lots of different kinds of applications. So where are we going from here? Well, we're in the process of developing a bacterial strain that we hope and we expect will make bigger proteins. We think we've got a way to keep it from choking on the large protein and make decent-sized proteins for us. We are in the process of making a whole new series of goats. The old ones are still going. I was up at 6 a.m. yesterday morning out in the barn. We started kitting. We started kitting at the beginning of the week. So I was helping deliver a set of twin goats at 6 a.m. yesterday morning. And then we had another set yesterday afternoon after I left. So the other folks had to take care of that. So we have the whole goat thing going. We're working on the silkworms. What we'd like to do is, instead of adding ours to the mix, what we want to do is knock out the one they use and substitute ours in for it and see how that goes. So we can change, instead of having only 5 or 10%, we want to be able to put 50 to 100% silkworm silk and see if they can spin it. Because if they can, then we really have a large-scale sort of a system to be able to work with. We're working a lot, again, on spinning conditions to try to see sort of how we can make things even better, make the best proteins that we can, and also how do we make things different. So there are some cases where we want something very strong and not very elastic. And lots of people want to use this for bulletproof vests. And I say, you know, I'll guarantee you I can make a bulletproof vest that works. The problem is the bullet's going to stop on the wrong side of your chest. Okay? So if you want to see an example of that, Google and look for bulletproof skin and put art, because there's a band called Bulletproof Skin, and so you'll pick up a bunch of their videos. Because we worked with an artist, interestingly enough, so we took some of the silkworm silk that was actually pretty lousy silkworm silk that had been transgenic, made transgenic, and she had it made into a fabric with some people in Holland. They grew a layer of skin on it, compared shooting a bullet into that, compared to shooting a bullet into skin that was grown on natural silk. And if you watch it, what happens is the bullet goes straight through the silk. And our silk, what happens is that it goes into the ballistic gel, and the skin never breaks. So that's why I'm absolutely sure I can make a bulletproof vest that'll stop the bullet. You're just dead anyway. You just won't bleed, that's all. The advantage is there'll be external sign that you got shot because the skin's still intact, all right? So I mean, that's an example of we have applications where we don't want lots of stretch. We need high tensile strength. So we need to know how to adjust the system. How do we make more Legos in order to make the system work? And finally, I'd like to end with lessons from a spider. And I use this when I talk to grade school kids and high school kids and all of those. So I think there are some things that are important to learn. So first of all, we call it research for a reason. If it was search, you only do it once. It's research because you have to do it over and over and over again to make it work. But when you finally do, it really is an amazing thrill. I mean, I can't tell you that when I got the first sequence of the very first spider silk protein, I said, at this very moment, there are only 2 organisms, if you want to call it that, on the face of the earth that know what spider silk looks like. God and I. That's a pretty impressive feeling. Hasn't quite been the same for the ones since then, but each one of them has been a pretty impressive sort of a thing. I think I showed you again, biology uses simple features to make complex things. Legos, Slinkies, zippers. Simple things makes an incredibly complex sort of a material. And finally, evolution is always the winner. So that's why we're following what nature did, because nature has thrown away all of those things that didn't work and is only moving forward with those things that do. So if we can mimic what 150 to 300 million years of evolution has done, we've eliminated all the things that don't work because they're gone. What does work is there, and what we know is that for 150 million years, spiders have done it exactly the same way. So it's got to be the best way, because anything else would have been tried and has been thrown away. All right? So I'll end with this. So here's where we keep our spiders. We have special little cages for them in this room.
Spider Central, huh? Whoa! Not funny!
You're now an official Spider-Man affiliate. I'd be glad to take any questions if anybody has. Yeah. So the main one we've used is called the golden orb weaver. The official name is Nephelocavipes. That's the one you saw most of. They're great. They're available most of the year round unless there's a freeze or a hurricane down in Florida. We've cloned the genes for every one of them from Nephilim. Golden orb weaver. Then the spiders that we use locally, there's one called our Gaiape, another one called catface. The catface we know we found in Logan. There's lots of them in Laramie. Our Gaiape is also called a cross garden spider. It makes a little cross in its web. We have not found them in Utah, but we haven't looked very hard. To be honest with you, we keep spiders in the lab to show people because we've already cloned and sequenced them. We don't need spiders. We kind of know all there is to spiders, but people expect if you work with spider silk, you need spiders. So we keep a half a dozen spiders in the lab to show everybody and take around to schools and things like that. So, yeah. Yep. No, they don't. Well, at least the natural silk we've implanted in rats and in mice. And the FDA control is a polyethylene rod. So we put a polyethylene rod on one side and we wrap the polyethylene rod with silk on the other side. was absolutely no statistical difference at all. And I think part of it is that it's a lot like silk. Silk is very non-antigenic as long as you clean off what's called the sericin coating. So, it's certainly at least as good as and probably better than silkworm silk. Yeah. Yes. Yes. Probably at least tenfold higher from bacteria. I mean, the only way it would not be is if you could find a plant that already is doing fermentation that you could use, because the cost of making the fermentation tanks is extraordinarily high. So if you're starting from ground zero, it's going to be a huge expense. If you were to take one over, then you eliminate that expense. So the actual cost is more amortizing the equipment to do it rather than the actual production cost itself. For the goats, the cost is basically the same, minus the purification cost, roughly the same as producing milk. It's not that much higher. Obviously, the silkworms are the bottom line. If you can do with silkworms, you're talking $5 a kilo, because that's what it costs right now in India to buy grade 1 silk. Yeah. Conserved for 150 million years between every orb weaver. Every orb weaver looks exactly the same for every one of those silks. We have, we've got some ideas about how to do it. I think better only means whether you make it stronger or you make it more elastic. I'm not sure that we can do the combination better than they do. It's not that we're not trying, but I think what we can do is shift something to be either stronger or more elastic. I'm not sure we're going to be able to make it more elastic and stronger simultaneously. I hope we can, but I'm not sure that we're going to be able to. Again, evolution. I mean, the only difference is they have to be able to spin it from a water-based solution at room temperature and pressure, and we don't. So maybe we can change the protein so that it works under non-natural conditions and maybe use a different protein. Yes. Yep. Okay, so there are 3 major proteins that make up cocoon silk and silkworms. What we're going to do is knock one of those out completely and put ours in instead. So what we did was added ours to the mix as a separate gene. And so we used the same regulatory things, but obviously we're not as good as what was already there. So if we just knock that out completely and put one in, then we anticipate that that's gone and it's going to have to use ours instead. Yeah? Okay, so our thought process is to use a Chinese finger. So we actually make a weave with a hole so that you attach it on the ends and then we stitch it down so that as it tries to pull, it's actually going to contract and hold the ends together. And then what we anticipate is being able to actually put stem cells inside along the edge to actually drive repair rather than scar formation.
Dr. Lewis, thank you for a great presentation. We have a little something here for you. Your very own friendly Spider-Man outfit, Dr. Lewis. We thought it might be a good prop for one of your next talks.
Thank you.
Thanks so much. What a wonderful example of world-class research at Utah State University being done in ways that will impact society and things that we do worldwide. Our next speaker you have on your table there is in April. Dr. Lane Kopic will be talking about empowering African women to transform drought-ravaged communities. Another example from our College of Natural Resources, Environment and Society of research that will have worldwide impact. So we're pleased about that. I want to thank Cecile Gilmer. Cecile has put these together for us for years now and does a wonderful job. Thank you, Cecile, for making all our arrangements. You have Slinkies on your table. please take them to your office, take them home to your kids, your grandkids, and thank you again so much for supporting, coming out to Utah State University Sunrise Session, and thank you again to Regents for the generous sponsorship. Have a great day.